Hi, everybody. As the last few stragglers are coming in, we want to kick off Cosyne 2025, and it is my absolute pleasure to introduce you to the other member of the exec committee who's present here today, Anne Marie Oswald. Take it away.
Why, thank you, Stephanie Palmer. All right, thank you, Stephanie Palmer, the other member of the executive that is here today. I'm here, and our job as the executive is to open the meeting and to acknowledge all of the incredible work that was done by the general chairs, the program chairs, the workshop chairs, everyone else who put this meeting together while we watched. And I just want to say thank you all for being here. As every year, but this year in particular, I'm reminded of how much of a privilege it is to get together as scientists at meetings and to be able to discuss what we do and advance our world. And I think this is amazing. And so thank you all for being here. I'm so grateful for all of you. Let's be grateful for each other, and let's enjoy this experience. And without further ado, I'm going to introduce our two program chairs for this year, Kevin Franks and Tatiana Engel, who are going to give us a nice introduction to the meeting.
Bonsoir tout le monde. Bienvenue à Montréal. That's it. That's all I got.
Good evening, everyone. It's my great pleasure to welcome you all to Cosyne 2025. Over the past 20 years, Cosyne became a very thriving and large community. So here are just a few numbers. Today, we have over 1,000 registered attendees for the main meeting, and we have 778 attendees at the workshops this year. The program, which you will experience over the next couple of days, was carefully crafted through a peer review process. We recruited 60 program committee members, who then recruited 377 reviewers, which evaluated the abstracts. This year, we received 1,003 abstract submissions, which is a new record for a North American Cosyne. Out of those abstracts, I try to be positive. Out of those abstracts, the best 450 were selected for talks and posters with an overall submission rate of 45%. While these numbers are certainly impressive, I would also like to put them into perspective with a quote from Kwabena Boa, who used to say, "We are on an island. The mainland neuroscience is out there." So if you think about it, it's true. The mainland neuroscience is a vast landscape, rich in natural resources, such as glia cells and neuromodulators, dendritic spines, and all the molecules, with over 30,000 attendees coming to Society for Neuroscience meeting every year. Over all these years, Cosyne has carefully fostered close ties with its big neighbor. But recently, another superpower has emerged on the other side of the world. It is AI frontier, notorious for its fast-paced progress, fueled by corporate investment. Will Cosyne fade in the shade of these superpowers? Our answer is no, because Cosyne is strategically located right in the center of the world, so that the flow and exchange of ideas necessarily has to go through this tiny island. Questions which would be unthinkable on either of these large mainlands are freely explored by the Cosyne community. Cosyne innovators bravely and seriously ask questions like, is the brain a transformer? They also wonder, absolutely seriously, how do neuropeptides generate line attractors in the brain? But not only do these questions flourish here at Cosyne. Cosyne has also excelled in exporting ideas that have influenced the major empires. For example, just to give you one example, the state-of-the-art benchmark idea is now completely commonplace on the mainland neuroscience. And looking 10 years ago, in 2013, when Valerio Mansi discovered line attractors in PFC, they didn't even have a metric to tell how good their line attractor is. Look what happened over the years. Already in 2020, we had a line attractor score in WNCIT and precisely measured in hypothalamus. And this year, the line attractor score reached the state-of-the-art number above 2. And it keeps going. Who knows, maybe this Cosyne will be the meeting where the new state-of-the-art record will be set. So in the spirit of this international and multidisciplinary community, we selected an outstanding group of invited speakers who will take us on a journey all the way spanning from molecules, neuromodulators, developmental plasticity, to the theory of deep artificial neural networks. These talks, we arranged them in a program over the next 3 days, and this program will be framed by 2 keynote lectures. One will be given tonight by Max Bennett, and he will bring the algorithmic perspective to understand brain evolution. And another one will be given by David Anderson, coming from the mainland neuroscience and wondering about line attractors in the brain. The program is filled not only with invited speakers, but also with 420 posters and 30 selected talks, which we arranged in these sessions thematically fitting with the invited speakers. And now I will hand it over to Kevin, who will tell you how we organize the abstract selection process.
Thank you. Okay, so I'm going to get a little bit wonky here. So Tatiana mentioned this, and I'm going to go into a little bit of details. So we received 1,003 applications, right? We selected, we picked 60 members of a program committee who then recruited 377 reviewers. And these reviewers each reviewed multiple abstracts such that each abstract was reviewed at least 3 times. The abstract was scored and weighted by a confidence measure such that we took the top 450 abstracts. From these, the program committee were asked to score and vote for those that they thought would represent the best balance of diversity, being demographic diversity, subject diversity, levels of seniority, and geographic diversity. And with that, we came up with 30 talks. I want to talk very briefly about the reviewers, because these are people who really make this happen. And it was a surprise to me, at least when I put these together, that there is a really impressive balance of seniority levels for people who are reviewing your abstracts. And we'll get to that in a little bit. So here are the scores. These are the weighted scores for all 1,003 abstracts. And you can see that there's a normal distribution with a cutoff at about 6 for where we accepted the top abstracts. Again, we took the top 60, and from those we picked 30 talks. We also asked the people who submitted abstracts to self-report by gender and minority status. And based on those self-reports, we looked at the scores for both first and last author, and we were very reassured to find that there was no difference between white male, female, and minority status for both first and last author. But we thought that we could do a little bit better than just looking at these stats. In fact, this goes back several years to Cosyne 2020, when Haruka Ushida, who was a PhD student at the in economics at the University of Chicago, decided to do a blinding study. And so in 2020, each of the abstracts for Cosyne were reviewed 4 times. 2 of them were blinded, and 2 of them were not. And the paper in which this data is analyzed is now available, and it's called What Do Names Reveal. You can find it if you look for it. But I'm going to summarize some of the main findings. So, first, we asked, how does blinding affect scoring? If we take the scores of male versus female first authors, when they're unblinded, we found no significant difference between them. And when they were blinded, what little patterns there might have been were eliminated completely. That's very reassuring. Slightly less reassuring was that there was a significant, slight but significant difference between the scores of first author abstracts that came from students versus first author abstracts that came from people who are not students. This could be postdocs, fellows, faculty, or other people who have graduated. When those abstracts were blinded, we found no difference. That's something to think about. So this was from 2020. Now we have 5 years, and we can go back and look and see what's happened in the meantime. So what we're showing here is the blinded score on the x-axis, or the ranked blinded score on the x-axis, plotted against the score of the non-blinded abstracts. And you can see that for male and female, there's no difference. And if we look 5 years later at the number of citations for each of these abstracts, once again, there is no difference between them. That's fantastic. What about this? Right? We see that student abstracts are, in general, weighted or scored very slightly worse than non-student abstracts. What happens when we look and see what happens to those papers 5 years later? That difference is completely eliminated. Gets even more interesting. What Haruka did is look at the institutions that the authors came from and used the US News Global University ranking to sort the elite universities from what I called the great unwash, right? The rest of them. And if you look at the scoring of these abstracts, you can see that when the abstracts are great, the abstracts that got really high scores, there was no difference between the elite and the rest of them. However, for the abstracts that had lower scores, the people coming from the top 20 institutions were given the benefit of the doubt. This, I think, also explains partly the difference between the scoring of the student versus the non-student abstracts, right? Perhaps when we know the authors, we tend to give them the benefit of the doubt when something's interesting, or something is unclear. What happens when we look at those abstracts 5 years later? Once again, there is no difference. I got a little excited there. I'm going to let Tatjana take over.
All right, so after all of this review process, as I told you, we got 450 abstracts, and now I will tell you how Cosyne this year broke the pillow rule. So those of you who are here at Cosyne for the first time may think that the pillow rule is a well-known rule in interior design which recommends how many pillows you should arrange on your sofa for it to look stylish. You are mistaken. At Cosyne, pillow rule concerns with the last author with the highest number of accepted abstracts, a record which has been held by Jonathan Pillow for years after years after years. So as you can see the data here, this year there were 2 PIs who got 5 abstracts accepted, a record which only accessible to Jonathan in the past. You may think one of them may be Jonathan. You are mistaken. Congratulations to Brent Doiron and Juliana Georgieva. And also, from the depths of my heart, I want to congratulate the entire biophysical modeling and neurodevelopmental plasticity community for breaking the pillow rule, finally. All right, from now on, I will take you a little bit on the logistics of the conference altogether, and also about specifics of this year's conference. All right, so as you know, the typical Cosyne timeline in the past years was to submit abstracts by the end of November, receive acceptance notifications in early January, and the conference was taking place in late February, early March. So what we did this year, we moved the abstract deadline 1 month earlier so that acceptance notifications can go out in December, and we moved the conference 1 month later to the end of March. So the idea behind it was that we would be giving people more time to get their visa process happening, and we hope it would help. We understand that it's always tricky to get all the paperwork in place to be able to attend conferences, but we are very interested to hearing your feedback, so please email us, let us know how this experience was, whether this additional time was helpful for you. So this is now our program, focusing on logistics. As you see, we didn't forget about lunches and coffee breaks. Those will be definitely on the program. And we will have 3 poster sessions. And again, take an example from the Cosyne last year. The poster sessions on Friday and Saturday will take place right after lunch during the day. It was an overall positive evaluation by the Cosyne community to have posters during the day rather than in the evening. So we continue with this year. And again, if you have any feedback, don't hesitate to email us. This frees the evening for special events. In particular, on Friday night, there will be a Simon's Foundation social, and everybody is invited. Coffee break on Saturday will also have a special Shanahan Foundation social, where you can meet Shanahan Foundation fellow and learn more about this program. And finally, on Sunday, there will be mentoring and networking lunch. This event is by registration only, so if you didn't have a chance to sign up yet, please do so quickly. All right, on Saturday afternoon, we will transit to workshops, and we didn't forget, there will be a Cosyne dance party. However, we learned from mistakes of last year. As you can see, the party is not scheduled on Saturday night of the main meeting because we want all of you to come back on Sunday morning. So the dance party will take place in the workshop place on the last day of the conference. Okay, how do you navigate and learn about all of these events and activities at Cosyne? We have multiple aids. In particular, Hoover app is the official Cosyne app where you can find the full program, arrange your own personal schedule, read all the organizer announcements, and also engage in community talks. It's not the only app you can use. There is another app called Axie, which you're welcome to download. It's also very intuitive to use. If you like an abstract, sweep to the right. You don't like it, sweep to the left. I guess many of you are familiar with the process. And as you do so, the smart AI system at the back end learns about your preferences and will suggest the abstracts you're actually gonna like. So this app also has extra features, such as you can also find your poster buddies. So you can actually get in touch with people who have very similar poster preferences so that you can go together to the poster session. All right. In addition, we encourage all of you to use Worldwide Nearer, a platform where you can upload video and audio recording explaining your poster. And the advantage of it is that every Cosyne poster will then get a permanent DOI, so that even after the conference, people can reference and look up this work. So please upload your posters, videos, and audio recordings to Worldwide Nearer. In addition, nothing can work nowadays without AI tools. Obviously, Worldwide Nearer also use large language models, which can digest all the abstracts for you. If you're too tired to use the entire 300-word abstract, Worldwide Nearer presents you a short abstract summary of an abstract, like who needs that? In any case, and moreover, it also suggests you questions to ask to a poster presenter. So poster presenters, if you notice that you get the same question over and over and over, you can guess what is happening.
All right, I'm going to end these intro remarks with the best part, which is thanking people for making this happen. So first, I want to thank the executive committee, Anne-Marie, Stephanie, Alex, and Tony, who aren't able to be here. This meeting is your baby, and it wouldn't happen without you. So thank you for the opportunity to let Tatiana and I put a show on for you guys. I want to thank Bing and Chand, who were the, sorry. I want to thank Bing and Chand, who are the general co-chairs, and they really shepherded the two of us through this process. There's a lot going on, and we came in sort of wide-eyed and terrified, and they assured us, they told us how to do it, and they were there whenever we needed them. So thank you for that. We had nothing to do with the workshops, but this is, in the general meeting, will be the only time that all of us are in one room together. And so I want to take this opportunity for us and for you to thank the workshop chairs, Suyong Chung and Guillaume Lajoie. In addition to that, there were several other chairs who basically made sure that everything happened. And I particularly want to thank the sponsors who have really, allowed us all to be here today. So thank you for their generosity. So we mentioned that there were 1,003 abstracts that were reviewed 3,000 or more times. That is a huge amount of work that was done by an army of volunteers of 60 program committee members and 377 reviewers. So I'm going to ask for 15 seconds of applause. And finally, we both are deeply, deeply indebted to Leslie Weeks and to Tomasz Hromadka. Leslie runs this conference. She makes everything happen. Thomas also makes everything happen. These two have been there for us at every moment. Emails at the middle of the night. Something's broken. Somebody can't come. Whenever there's a problem, you might think that we've solved it. We haven't. Leslie and Thomas solved it. And so thank you for all your efforts. And so with that, I want to introduce tonight's speaker, the keynote for tonight. Well, the keynote speaker tonight is Max Bennett, and he will tell us about the 5 breakthroughs in the 600 million year evolution of human intelligence. A useful first approximation. So a little bit about Max. He graduated from Washington University with a degree in economics and mathematics. With that, he then founded several AI companies and got interested in neuroscience. Started reading and thinking deeply about this. And with an outsider's perspective, wrote what one of the executive committee members says is that possibly the best neuroscience book I've ever read. So without further ado, let's welcome Max Bennett.
All right, thank you very much. So I want to start by acknowledging I am definitely an outsider. So I am particularly honored. I'm not an academic, so I'm particularly honored to be here. However, I just found out that last year, from a friend, last year the person who was at this moment was just showing bee videos for like an hour, so maybe I shouldn't be so honored. No, I am. And it's particularly full circle for me because a lot of people that inspired this book are here, Paul Shizek in particular. This whole book that I wrote wouldn't have been written if I hadn't read some of his work. So I'm very much indebted to him and a lot of people. So, okay, let's jump in really quick. Background on me, my whole background is in commercializing machine learning technology. I won't bore you with the details of products and all that boring stuff. But it was here where I really became fascinated about 6 years ago, where I think a lot of people in this room maybe found it in a different way, with sort of Moravec's paradox, which is, why is it the case that AI systems can do certain things that brains can't, and why can brains do certain things that AI systems can't? And I want to ground the sort of adventure back in time that I want to go through in a particular sub-case of the question that I think everyone here asks, which is, we all want to understand how the brain works, but I'm particularly interested in this middle layer in Mars's 3 levels. So it's just important to ground what my North Star question is as we're adventuring back in time. I'm really interested in what are the algorithms underlying how the brain works. I'm less interested in the underlying implementation details. And so for 80 years, the standard paradigm here, as we all know, is functional decomposition. And so we have a set of functions that we know the brain does. We have all these regions, and we've spent a ton of money and time trying to create mappings between these things. And I think, as most of us know, at a computational neuroscience conference, there's at least 2 very large challenges with trying to understand the brain by doing this. And this is the first sort of block I ran into when I began my own personal journey reading a bunch of neuroscience textbooks, which is, 1, functions are highly distributed. So language doesn't actually live in one area. Visual object recognition doesn't live in one area. Decision-making doesn't live in one area. And then 2, regions do multiple functions. Dopamine doesn't do one thing. Motor sensory signals are overlapping in many neocortical regions. A better approach, which obviously there's a ton of really fascinating work in, is instead of focusing on functions of regions, we can shift the question. We can ask, what is the underlying algorithm implemented in a given region that might be used in a variety of different functions? We can ask, what is the underlying algorithm implemented in the neocortical microcircuit, for example. But even this is not a panacea, because we don't have the required engineering to actually go into even a neocortical column and come out and say, this is the algorithm being implemented. So the entire argument underlying the book is in some ways relatively simple, is that there's an additional tool, not necessarily the best tool, but an additional tool in our toolbox that tends to be underutilized, and that is evolution. So we can ask this question in a slightly different way. We know that the human brain was not designed from scratch, it evolved. I'm going to assume that everyone here believes in evolution. And because of that, evolutionary steps are constrained and path-dependent. And we know, and thus, knowing the prior steps gives us information on the subsequent steps. So the path is informative as to the structures that ended up emerging. So it gives us this other alternative that can be a useful tool, which is what would happen if we went back to the very first brains, to the extent that we understand how they worked, and tracked the modifications and subsequent intellectual faculties that emerged over time. And that's the whole idea. And there's already a ton of work in trying to come up with evolutionary frameworks. Credited, the evolutionary story is wrong. The brain did not evolve in adding layers. Reptiles have a limbic system. It has the same problem as functional decomposition. The emotions exist in the neocortex. Lots of cognition happens in subcortical areas. It's not grounded in the modern view of comparative psychology. And most importantly, it tells us nothing about the algorithms. Nothing. So we need a new evolutionary framework, one grounded in up-to-date evolutionary biology, comparative psychology, and based on a modern understanding of what actually works in AI systems. Okay, now what's really cool is we actually already know or have a ton of information as to the key neurobiological changes that occurred in the human lineage. And so there's a ton of comparative neurobiology work that has done this. I won't read this, but we have a good understanding as to where in our phylogenetic tree certain neurobiological changes occurred. The key question, of course, is what was the adaptive benefit of these changes? What capacities did they enable, and what algorithms were actually implemented by them? And so to do this, we have to take the comparative approach and apply it to behavior. And so the data here is sparse, but we can do this. So here's just an example of the comparative approach applied to behavior. If we wanted to argue that a specific behavioral ability emerged at this sort of, I don't know if you can see my mouse, but this sort of dotted circle area in our phylogenetic tree, You can make this argument with two conditions, two main conditions with maybe a third. One is the in-group condition. It should be the case if let's say spatial mapping evolved in early vertebrates. It should be the case that diverse modern species of vertebrates are capable of this ability. Early diverging species of vertebrates can do this. And it should be causally linked to similar homologous neurobiological regions. And it should be a case in the outgroup condition that diverse groups don't show this ability. And if they do, it emerges from non-homologous neurobiological regions. And so you can apply this to various behavioral abilities. For example, allocentric spatial mapping is something that's been tested across many, many different species. I think most people know what this test is, but very briefly, you can have a grid of 5 by 5 little cups, and none of them are cued, and you can ask the question, can a fish remember one of these locations without it being cued, right? So if you put food in one of them, and you only have extra maze cues, it finds it, and then you do it again without the food in there, so there's no olfactory cues. Does it actually identify where it's located? Now you can do this. Diverse early diverging vertebrates solve this effortlessly, and it's very sparse results in invertebrates, possibly independent evolution in honeybees and cephalopods. So the parsimonious conclusion here would be the ability to remember allocentric locations in space emerged in early vertebrates. So we can apply this to a variety of different things. Now I have two caveats before we chart back in time. One is I am not making, the point of this is not to compare humans to all extant species. It is to draw the human lineage. So that means if one makes the claim in our lineage spatial mapping evolved in early vertebrates, that's not the same thing as saying only vertebrates have spatial mapping. So I'm taking a human-centered view. And then the second caveat is we don't have a lot of data on the same behavioral studies across species, right? So this is sort of, given the data we have, what does it suggest? Everything is pending us doing more comparative psychology studies on other animals. Okay, so if you do this, this is an example of the types of behavioral abilities you can start charting in our phylogenetic tree. When did these things seem to emerge? And then the big question is, can we infer anything? Are these all just a litany of random new neurobiological changes that enabled yet another sort of new behavioral ability? Or is there some relationship between these things? Is there sort of an underlying algorithm that was enforced by many neurobiological changes? And was this algorithm applied to many different behavioral abilities? And I'm going to argue yes. So let's go back in time and chart the story of the human brain. So we all know that the first animals did not have brains. It had a nerve net. So by the time the first metazoans appeared, there were muscles, there was neurons, but there were nothing we would probably call a brain. They were most likely akin to today's cnidarians. All animals today with brains evolved from our common ancestor, the first bilaterian. Within the first bilaterian, it's believed that there was the first brain, and most notably what makes them distinct is bilateral symmetry. And it is not a coincidence that pretty much all land locomotion machines are bilaterally symmetric. It is simply the best body plan if in a two-dimensional plane you want to navigate. If you wanted to navigate with a radially symmetric creature, you'd have to have some motor apparatus to move in any direction at once. With a bilaterally symmetric system, you can simply have a motor system for moving forward and then something much simpler for turning. So the first bilaterians were clearly the first animals, or very likely the first animals to actually move around. And so to get a window into what was its first brain doing, and what drove the evolution of this first brain, we can use various model organisms, C. elegans being a great one. How does a nematode find food? Well, we all kind of know that it uses taxis. It's a very simple algorithm that does not require having any rich understanding of the world. You simply keep going forward. If a good thing is increasing, so you're climbing a gradient, you turn randomly if it's not, or if it decreases. So that's clearly how nematodes find food. Now, if you go into the brain of C. elegans, obviously it's much messier than this, but in principle, it's actually quite simple as a navigation machine. There are sensory neurons that detect the presence of good things, like olfaction, or smells, chemicals emerging from food, and then there are things that detect bad things, things you would turn away from, temperatures above a certain threshold, so on and so forth. What is interesting about simple bilaterians is sensory neurons are not really distinct from votes for a certain movement. So sensory neurons directly detect valence. So a lot of the sensory neurons that are responsive to chemical cues for food are also sensitive to an animal's internal state. So you could cast a lot of these sensory neurons as votes for turning or forward locomotion, as an example. And you need a brain because you need to make trade-offs. If you're spending a lot of energy moving, then you need to be able to integrate the votes for turning or forward locomotion into a single place. So you can do lots of experiments with simple bilaterians and force them to make trade-offs. Put a nematode in a petri dish, put a copper line in between, which they find aversive, put an odor on the other side. Does the nematode cross the line? Depends on two things: the relative strength of the odor to the copper and how hungry they are, right? So they're clearly making these types of trade-offs. Now, the question is, cool, the first brain did taxis, that's not particularly interesting. What other things emerged in the first brain can be seen through the lens of a machine designed for taxis? In reality, gradients are not as simple as they are in a petri dish, right? So in reality, smells are noisy. If I detect a smell in one moment, I might not detect it in the next moment, even if I'm moving in the direction of the food, because there's currents and all of these things in the open water. And so I think a good analogy is to the Roomba. I don't know if anyone has a Roomba, but modern Roombas have something called dirt detect. Does anyone know what dirt detect is? Dude, yes, that is awesome. No one's ever said yes to that. Okay, it is a window into the beginnings of the brain. What dirt detect does is you would think a Roomba would just move around randomly or have some sort of random walk to clean up all of your food or all of your dirt, or if you're throwing food on the ground. But what it actually does the second it detects dirt is it changes its entire behavioral repertoire. It starts doing local search. It stops, slows down, and starts turning in a circle randomly to see if it finds dirt again. And it does this for a very simple reason, which is the world is clumpy. If I just detected dirt, it's likely there's dirt nearby. So nematodes do exactly the same thing. So nematodes and other simple bilaterians like planarians have behavioral states. If a nematode detects food, it floods its brain with dopamine, and it will change its behavioral repertoire from roaming to dwelling. It'll do some local search until dopamine levels drop, and then it will shift back to roaming. Very, very similar to dirt detect. And what's interesting is you could kind of cast this as like what one might call the navigational theory of the origins of affect, which is, what is affect? It's a two-dimensional grid of valence and arousal. And when you look into a nematode, this I found beautifully fascinating. Dopamine neurons are sensory neurons in C. elegans. They detect food around the animal. And there are two groups of serotonin neurons, but one group detects serotonin in the throat of a nematode. It detects the presence of consuming food. So this is the basic separation between dopamine and serotonin, although clearly elaborated through evolution, hold the very basic template that we still see today, which is dopamine is a nearby, there's something good nearby, sort of exploitation chemical, and serotonin is this sort of consumption, something good is happening chemical. And we see this even in the basic sort of navigational states of a nematode. Cool. Further, bilaterians can learn associations. I'm running out of time, so this we know. So they can learn, obviously, if you have a system that needs to make choices that are energetically expensive to navigate, it's usually very beneficial to learn, should I still turn towards this thing? So you can teach them to change their valence. Now, what I think is cool is the second you have a system that needs to learn associations, you have a problem, which is how do you solve the credit assignment issue? So for example, if I tap the side of a slug and then it gets shocked, the tap was not the only thing in the animal's sensorium that happened in conjunction with the shock. It's detecting lots of things around it. So how does it choose the predictive cue? All of the basic things that we see in Pavlovian learning across mammals, many of them, with maybe the exception of latent inhibition, we see in very, very early bilaterians. These are all kind of simple tricks to solve the credit assignment problem. They're clearly not perfect, but they're good rules of thumb. The thing that happened right before the outcome is probably something I can use to be predictive of it, as an example. Okay, so this is the beginning of breakthrough 1, but the idea here is that a huge suite of things that emerged in the first bilaterians, from categorizing the world into good and bad, bilateral symmetry, behavioral states, associative learning, can all be seen as different things that benefited taxis navigation. It was all about, how do I change my location? Okay, so this is our little grid. Obviously, the descendants of the first bilaterians proliferated broadly. There's a huge Cambrian explosion, predator-prey feedback loop. What happened with the first vertebrates? And within the first vertebrates, we see the basic template of even the human brain. We see a forebrain, a midbrain, a hindbrain. We know this from the lamprey fish. We see all the familiar sensory organs emerge. Fish, maybe in this room, a lot of people have studied fish, but a lot of people are surprised that fish are actually quite smart. They can learn to swim through mazes. They remember that a year later, you can teach fish to jump through hoops. You can teach fish to push buttons to get rewards. They can learn the exact timing between events. You can even teach fish to identify an object in one shot when rotated in 3D space, even controlled for color, something even modern CNN struggle with. And so what happened in this first vertebrate? And obviously fish I'm using as a model organism because they've been studied most extensively. But so the basic macro structure of the cortex, thalamus, and basal ganglia we see even in the lamprey fish. So all of the things that we have associated with the basal ganglia being an actor-critic system, for example, all of the neurobiological components that we think of as engaging in an actor-critic system are present within a lamprey fish. So it seems very likely that this basic macro structure emerged back then. And I assume most people are familiar with TD learning and all of that good stuff here. Yeah, so I'll probably skip over this because I'm guessing most people are familiar with this. But the key thing is what Schultz found in monkeys, which we typically think about as what exists in mammals, which is dopamine being a TD learning signal. In other words, tracking expected predicted reward from a given moment in time. We have every reason to believe actually evolved in early vertebrates, not in early mammals. One, the overall structure surrounding the dopamine system is the same. Two, we see these same TD learning signals in homologous brain structures, even in teleost fish. And then three, the behavioral abilities that you would expect from something that can engage in temporal difference learning, you see even in widely diverging and early diverging species, such as teleost fish. A good example here is omission learning. So the people a la B.F. Skinner always had a problem with omission learning because in order to learn from omission, you have to find something that didn't happen rewarding. So suppose you have a fish in a box, and if it doesn't swim to the other side, you give it a shock. It'll learn to swim to the other side. Fish do this effortlessly well, and it will continue swimming to the other side whenever you cue that in 10 seconds is going to be a shock. But the B.F. Skinner approach, this is confusing that this behavior continues because there's nothing rewarding. It just continues to do the behavior. And in fact, if you take a cue and you show it the cue when it successfully avoids, a fish will find that cue rewarding. And what that this was a problem, a while ago, which is like, how can that actually be rewarding? From the lens of TD learning, this is trivial. Of course it's rewarding, because if I expect something bad to happen and it doesn't, that is a positive TD learning signal. And anything that happened in that moment would inherently be reinforcing. So it will continue to be reinforcing if I continue to expect that the trial start is going to shock me. And the point is that fish do this effortlessly well. So what seems to have happened is dopamine, which began as this fuzzy average of recently experienced reward, can be repurposed into a precisely computed predicted future reward signal. And per Joshua Burke, who was recording extracellular dopamine in the striatum, these rapid changes in dopamine levels are effectively your TD learning signals. So it makes sense that dopamine would have been the chemical that was repurposed in this way. Okay, now the key thing about TD learning that makes it so amazing is it can enable sophisticated behavior without any machinations for planning. And so intuitively, it makes sense that if you're going to have reinforcement learning evolve, it would start in a model-free way without all of the necessary machinations for how to pause, render possible future paths, and then select one. The question is, okay, even if one accepts that it seems to be the case that TD learning emerged in early vertebrates, why do we get this sort of explosion of sensory organs and all the other things that emerged with early vertebrates? Well, TD learning needs three things. You need a base reward signal, which evolved in early bilaterians. You need a mechanism for generating learning from those TD learning signals, which seems to be what the basal ganglia was doing. But you need a mechanism for recognizing states in the world. It's not useful to have a machine that can learn to execute arbitrary behavior through trial and error in response to a state if you can only recognize 3 states of the world. It's useful to have a sort of a complex sensorium. So you get this sort of feedback loop between all these things. The more sophisticated your TD learning can be, it applies more pressure for more sophisticated pattern recognition, and then vice versa. The more sophisticated your pattern recognition machine, the more pressure there is for more complex sensory organs. So these are all sort of mutually enforced. So you can see, it's not surprising that also in arthropods, you seem to see a similar independent emergence of similar types of complex sensory organs in non-homologous ways, so on and so forth. Okay. Now, early bilaterians had no, in the classic sense, pattern recognition. In other words, C. elegans doesn't recognize things in the world based on the pattern of activated sensory neurons. It recognizes things in the world because a single sensory neuron gets activated. In fact, C. elegans seem to end up in a state where a single sensory neuron has protein receptors for many, many different things, because what really matters is this thing drives turning or this thing drives forward locomotion, not what in particular it recognizes. Of course, early vertebrates, we see a pallium, and the pallium is clearly a pattern recognition machine. In fact, in early vertebrates, we see the portion of the pallium, which is homologous to the hippocampus, that recognizes places in space, seems to do the same thing. So if you lesion that section of teleost fish, they also lose the ability to engage in allocentric spatial mapping. So the medial pallium and lamprey seems to do that. Okay, so one final point is, the way we learned about CNNs was the neocognitron, which was inspired by the sort of hierarchy you see in the dorsal pathway and the human primate neocortex. And this is how we sort of thought that vision works, or we think vision works. What is interesting is a fish eminently recognizes things in the world, eminently recognizes things when shifted in 3D space in one shot without any clear hierarchy. So it's an interesting place to look for an alternative algorithm here. Somehow the relationship between the thalamus and the pallium or the tectum enables this type of one-shot 3D object recognition without any clear hierarchy in this sense. So there's something interesting going on there. Okay, so as a basic schematic, again, first approximation, super oversimplified, but still as a first approximation, you can kind of think about this transition as you started with this basic steering brain, which cast everything through the lens of turning or forward locomotion. It was just for valence. And then you can build a reinforcement learning machine on top of that. You need the first step for the second step. Cool. So then, fast forward hundreds of millions of years, and the main thing that changed in mammals, obviously, is the emergence of the neocortex. And we can ask this question, why? What drove neocortical evolution? Now, if you look at what the neocortex does today, it gives you an answer that's, I would argue the evolutionary story suggests is probably not right, because it does a lot of things in the human brain today: visual perception, auto perception, motor control, executive function, language, so on and so forth. But there's a few wrinkles with this. Fish already had, early vertebrates, if fish are any signal of this, and lamprey are any signal of this, already had great motor control, already had great visual perception. So spending the extra energy on getting 6 layers of V1 is questionable. What was the adaptive benefit of that if fish already can recognize things just fine? The first mammals didn't even have a motor cortex, so it's clear that whatever the neocortex evolved for didn't seem to be motor control related. So what are the things, if you look throughout, if you do a meta-analysis of the literature, that seem to be unique behavioral abilities that emerge in mammals? So I'm going to list at least 3. One is this idea of vicarious trial and error. So David Reddish's lab has done amazing work on this. For anyone who's not familiar, these are great papers. So, Tolman identified as in the 40s that rats at choice points would pause and look back and forth. Now, of course, that doesn't prove anything. He hypothesized what they were doing, was he named it vicarious trial and error. They were imagining possible future states before deciding. They were training themselves vicariously and by imagining themselves take action. David Reddish, by recording place cells, found that at these choice points, when they do this sort of head nobbing behavior, they would see place cells actually do these sort of forward paths in each of those directions. Highly suggestive that mammals are engaging in a type of imagination. Maybe not imagination as rich as a human, but to the extent that they're imagining possible future states, certainly the evidence suggests that mammals can do that in a highly neocortically dependent way. He also did he looked at counterfactual learning. I love this study. They call it Restaurant Row. They put a little rat in this little circle, and at each sort of point, they have a choice between turning right to get one of the 4 foods or to keep going forward. And one of two tones is played that either suggests I can get the food right now in 1 second, or I'm gonna have to wait 45 seconds. So what this presents is a series of irreversible choices, because once I pass the door, I can't go back. Now what's interesting is if they know, just based on their preferences, that I prefer banana, and I get a tone for 1 second for cherry, and I decide to go for the banana because I want banana, and I get 45 seconds, the rats show all the sign of having regretted their choice. They pause, they look back. If you record their OFC, they re-represent the cherry as if they were actually pausing to imagine themselves eat the alternative choice, and they change their behavior the next time around. They're less likely to actually pause. So this idea of counterfactual learning, in other words, when something doesn't go your way, you're not just engaging in this model-free TD learning, you're actually pausing and rendering past alternative actions you could have taken. Evidence suggests that mammals can do that. And then this episodic memory is a charged term, so I'll call it episodic-like memory, but this idea that a rat can render past events, lots of good evidence for this. My favorite study here, I won't go into all the details, is you can make it such that for a rat to know whether to go right or left in a maze, the only way to know the right answer is based on something that happened several minutes prior. And so the maze itself is equally paired with right and left. The only way to know is I have to sort of remember some event that happened prior. And rats can do this. If you inhibit their hippocampus relative to any other place they inhibit, they fail to do it. So this is a hippocampal dependence behavior, suggestive that the sort of complementary learning systems theory is maybe onto something. So the idea here is, again, a first approximation. All three of these abilities can kind of be seen as different applications of one thing, one algorithm, which is just being able to render a state of the world that's not the current one, which is, of course, very consistent with our typical approach to understanding the neocortex as a generative model, being able to render things from one's own latent representation. Now, interestingly, you could take this and then ask, well, what are other things that mammals are uniquely good at? Well, mammals seem, relative to our ancestral amniote, to have much better motor control. If you compare, there's some really funny papers on people who have tried to answer the question, do lizards actually anticipate their movements, where their arms are going to go? Or is it really just a reflexive movement? And most evidence suggests they're not actually anticipating where their arms are going to go. It's a reflexive response to just the stimuli they're receiving. Whereas clearly a cat does that effortlessly. And so if we think about the common sort of view that motor cortex is this locus of motor control, the evidence for this in humans and primates is, is self-evident. Lesions cause paralysis, activity correlates with movement, stimulation causes movement. But all of that's unique to primates. So if you do that in a non-primate mammal, you don't see those effects. So lesioning the motor cortex of a rat does not cause paralysis. It does cause the inability to learn new motor skills, but retains the ability to execute previously learned ones. And it does cause impairment with highly complex tasks. So what this sort of suggests, again, not irrefutable evidence by any means, but it's suggestive that this idea of simulating future states also applies to motor control. What the motor cortex originally was doing was simulating specific body movements for the purpose of mentally training yourself, for the purpose of planning, and then later, when there's a direct motor cortex to brainstem projection that you see emerge in primates, then all of a sudden this sort of system originally designed for simulation can now also do direct motor control. So the evolutionary story can be useful in these debates between the function of things. Okay, so the idea, the suggestion from the evolutionary story is that the adaptive value of the neocortex is that it enabled animals to simulate. The key thing of tracking this evolutionary story is you can ask the question, not what does a neocortex do, but ask the question, what did adding a neocortex to this complex system that is the brain enable? So of course, this model-based reinforcement learning was repurposing lots of other older systems, but what was missing was an ability to have a latent representation of the world that can be rendered internally. Okay, two other fine points that I'll just make on things. It's clear or strongly suggestive that early vertebrates had some form of a cognitive map per their ability to remember locations in space. So if the sort of continuous behavioral evidence is suggestive that this story holds, then what it would suggest is that the cognitive map originally evolved not for planning, but for generating sort of homing vectors to a location in space. So remembering a location in space does not require me to actually execute planning possible future states. I can just remember if my goal state is X and my current state is Y, what's the homing vector between them? And just directly compute that as an example. The other interesting thing is the evolutionary story is nicely consistent with the sort of CLS theory of hippocampal function, where the beginning medial pallium was just for encoding allocentric locations in space, and mammals, part of that can be repurposed to directly look up and sort of merge representations in the neocortex. Again, just it's consistent at least with that story. Okay, so super oversimplified schematic here, but as a first approximation, you can kind of see brain evolution B adding this mental simulation of world on top of this previous model-free reinforcement learning system. Okay, so as we will accelerate as we're getting towards humans, what emerges in primates? Primate brain obviously scaled up dramatically in size. For the most part, what's new in primates is the granular prefrontal cortex and then these areas of sort of posterior cortex that are highly implicated in social behavior. And so there's still lots of controversy as to which of these abilities emerge where, but for the most part, I would argue that most of the evidence suggests that in early primates, you see theory of mind-like function emerge. This is one of my favorite papers on this, where you can do a Sally Ann type experiment on a macaque, and they demonstrate the ability to have a gaze bias where the person would think it is, even though they know the item is not in that box. And that goes away if you inhibit the uniquely primate granular prefrontal cortex, or a subregion of it in medial PFC. We also see in primates really, really good imitation learning per the tool use in primates. And this is the least well studied, but there is some evidence to suggest that primates are uniquely good at anticipating their own future needs. So for example, they've done, this is a study where they would have a monkey have to choose between a low treat and a high treat option, where these treats make you thirsty. So they're dates in the case of the monkey. And monkeys will learn to take the low treat option today for getting more water afterwards, whereas a rat will not make the same choice when you baseline the amount of food that you're giving them in thirst to make sure that it's the same amount of thirst that you're inducing. The common counterexample is like, well, mice hoard food before the winter. Doesn't that suggest some future need anticipation? But they've shown that that's genetically hard-coded. So you can just lower the temperature of a rat that's never experienced the winter, and they'll start hoarding things in a cage. So the idea here is what emerged. The underlying algorithm here was mentalizing, which is rendering a model of one's own inner state and being able to simulate different situations and ask what would actually occur in those states. And that could be applied in each of these cases. So theory of mind is obvious because modeling one's own mind or modeling the mind of others, mentalizing theory of mind are synonyms almost. But why do these matter for imitation learning and anticipating future needs? We actually have done this experiment in AI. So does anyone remember? I guess, yeah, Nice. So people remember the Alvin self-driving car stuff. So just a quick, for folks that don't know, this was the basic idea of imitation learning. What happens if I put a camera on a car and I drive around and I try to create a direct mapping between what the car sees and the angle of the steering wheel, and I try to train an AI system, a basic neural network, to just imitate me. Why does this fail? It fails because it never sees a human recover from a mistake. So the second something goes a little wrong, it doesn't know how to recover because it's only watched an expert. So there's many approaches nowadays, but one of the state-of-the-art ways to get around this is inverse reinforcement learning. You don't directly copy the expert. You try to model the reward system of the expert. You try to understand what they're trying to do, and then you train yourself with the inferred reward function. This is effectively mentalizing. If I want to learn and copy the way that you are using a tool, it is very useful for me to infer what you're trying to do so I can mentally train myself to do it, as opposed to just directly copy the exact behaviors I observe. So this is why it might be the case that improved mentalizing abilities had the emergent property of enabling really good imitation learning that we see in primates. And this is the famous first application of helicopters going upside down. Okay, why would it be important for anticipating future needs? So this is not me at all. Thomas Suddendorf came up with this idea in the 90s, which was this idea of maybe it would be the case that the same machinations that enables me to ask a question about someone else's mind, in other words, a disjointed mental state than my current one, would also be useful for asking about a future state of my own mind. So, and a simple example is it really any different, algorithmically, to ask what would James want after not eating for 24 hours and be able to answer that question versus asking what would I want after not eating for 24 hours? These are in principle not really that different. So this might be why once you get theory of mind, you get much improved performance on tasks that require me to anticipate a state of the mind that's not my current one, which we seem to see in all of the anecdotes of primates bringing tools to distant parts and prepping for cold weather and all that stuff. Okay, so the idea here is mentalizing being this new ability, perhaps is underlying a lot of these new abilities that we see in primates. And then lastly, this one will be the easiest. We see an explosion with humans. There's tons of literature on what happened uniquely with humans. I'm going to stick with what I think the evidence is most suggestive of, which is really language is what makes us unique. And it was not, a naive, I first thought the naive thing was like, oh, I'm assuming what you evolved in humans was Broca and Wernicke's area, and then the problem was solved. That's absolutely not the case. Broca's and Wernicke's area are eminently present in all primates. Wernicke's area in non-human primates is just the area that is engaged in mentalizing. And that's perhaps a clue as to what the origin of language really is. It seems to be that we just repurpose, at least in the posterior neocortex, mentalizing regions for this purpose of language. And so the most convincing theories of language evolution that the ones I find most convincing are that really what evolved is a unique learning program for language. So children have a learning program that makes them engage in joint attention. So it's very hard to get a chimpanzee to do this, to have an object that you're both attending to and confirming that both parties are attending to the same thing. That's the foundation of declarative labeling. If you have cues to ensure that someone else is thinking about the same thing, then you have a grounding for labeling things. Even in all the experiments that have tried to teach non-human primates language, it's very hard to tell whether it's an imperative label or a declarative label because the way they're trained is they say things and they get treats for them, right? So there's a lot of controversy around that. And then, of course, there have been lots of experiments that very young infants engage in a basic turn-taking before they even have the actual words for language. Okay, and then why would mentalizing be a prerequisite for language? Well, we know this eminently in AI at least, or at least there's the sort of famous paperclip problem. I'm guessing most people are familiar with it, but in case you're not, Nick Bostrom has an allegory of, imagine we have an AI system and I ask them, hey, can you please just maximize production of paperclips in this little paperclip factory, and that's all you do, and it begins to turn all of Earth into paperclips. And the point is that language does not well specify our requests, right? Language is clues, and then there's a constant inference process on both parts, trying to infer what did the person mean by what they said. That inference process requires, in a lot of cases, or at least it seems to require, mentalizing. I'm inferring why would you say this, given my understanding of what you know and what you think. So mentalizing is very useful for language. Okay, so the idea here is as a first approximation, just fuzzily looking at the data we have today, we can kind of see an arc that helps cast a lot of the changes, of course not all of them, but a lot of the modifications over the last 600 million years, really through at least 3 very key algorithmic breakthroughs. And each one clearly was dependent on the one that came before. So as an example, you start with reward. You can't have a model-free reinforcement learning system without some grounding in things being categorized into good and bad. And so with a basic taxis navigation system, we had, in early bilaterians, you had this grounding that could repurpose dopamine into this reward signal. Now you can't have model-based reinforcement learning without some mechanism by which you're rewarding things in the first place. So with TD learning, it makes sense that after that, you would get something that actually renders a model of the world and can imagine possible future states. In order to have a model of one's own mental simulation, you need the mental simulation in the first place. If I'm going to ask the question, what is someone thinking about, it's not a well-formed question if the other person is not in quote-unquote thinking. So mentalizing requires simulation. And then for reasons we just talked about, language is at least very benefited from having a system of thinking about other people's mind states. So the last few things, I'll just talk about why I think this is interesting or useful. In some way, you could almost cast the evolution of the human brain as just going on the whole AI train of more data is more performance. By which I mean, reinforcement in early vertebrates was finding a new source of learning, which is the outcomes of your own actions. So you're learning from your own actual behavior. In mammals, the aperture of data on which you could learn expands. Instead of just your own actual actions, you can also learn from your own imagined actions. So a completely new data source. When I reach a choice point, I don't have to wait for the actual behavior to learn. I can imagine doing possible things, and in my mind's eye, learn from the outcomes. So that's expanding the scope. With mentalizing, in other words, being able to infer reward functions from other people, further expands that aperture. Now I can learn from others' actual actions. I can watch what they do and infer why they might be doing it and try and replicate their behavior. And what's so magical about speaking is you can learn from others' imagined actions. So in a tribe, I could come and say, I'm imagining this plan of how we're going to catch the boar over there. Let me share the outcome in my mind, and then we can go learn together from what was in my mind. So one way to cast all of this is just expanding the aperture of data on which, of course, again, a simplification, but I still think an interesting one. So the idea from 10,000 feet is that many disparate intellectual skills emerge from common algorithmic breakthroughs that built on top of prior algorithmic breakthroughs. And I'll just give I'll close with just 4 reasons why I think this is not just sort of an interesting intellectual exercise. I think there are 4 ways in which I think it can be useful. One, I think it helps interpret the brain as a whole instead of with functional divisions. So we can ask questions like, what ability did adding a neocortex enable? Instead of saying, like, what does a neocortex do? And it enables us to incorporate the rest of the system that something evolved in and not have to be functionally divided the way a human designer would. It helps add constraints to the interpretations of functions, so it challenges things like motor cortex evolved for motor control because the evolutionary story is not consistent with that. It narrows the classes of algorithms to evaluate, so it suggests that the story of the neocortex in some way being some form of generative model is consistent with what we would expect given the evolutionary story. And it helps explain these multi-purpose neurobiological features. This, the grand debate classically that is dopamine a wanting signal or a learning signal? Cast through evolution is almost a silly debate. Of course, it's both. And of course, it started as one thing and got elaborated to do many, many different things. And we can make sense of it through the story of evolution. And that is it.
All right, so that was fantastic. We have a little time for questions, so I'm going to ask if you do want to ask, well, people have already gone to the microphones. Let's go one at a time. Let's start over there, please. Could you also introduce yourself, say your name and where you're coming from?
Sure. Hi, good evening. Karen Moxon, University of California, Davis. Thank you for your talk. I wondered, can you hear me now?
Yeah.
Okay, sorry.
I don't know where you are, though.
Over here.
Okay. Yes, hi, sorry.
Karen Moxon, University of California, Davis. Thank you for your talk. I was wondering, when you talk about mentalizing, do you include empathy in that? Do you think that empathy is kind of a negative side effect or one of the benefits that have given us today's human capabilities? And if you think it's a benefit, do you think it is something that should be included in the development of artificial intelligence or something we should leave out?
So the empathy literature is really interesting, because if we define empathy as the ability to feel, this is why I find the framing on thinking about algorithms so critical. If what empathy is my ability to feel someone else's feeling, there's multiple ways that could be executed. So one is I could have genetically hard-coded ways to feel what you're feeling when you feel it, right? And so there's lots of evidence that rats, for example, have that form of empathy. A rat in distress causes distress to another rat nearby, and rats will do work to help another rat to reduce that distress. It doesn't suggest, though, or mean that the rat is actually closing its eyes and imagining what it would be like to be the other rat and making a decision on the basis of it. So I think that's the type of empathy one gets from mentalizing, because I can put myself in your shoes and imagine why you might do something that I would not do. Whether that's useful for AI, I would imagine yes. I think, but we get that wrong a lot, right? So if the solution was let's just make AI systems have the same form of mentalizing empathy humans do, that would be insufficient given the power that we're giving to AI systems. Because as we all know, we don't always get it right when we imagine ourselves in other people's shoes and think we know what they want, right? So I think it's a useful first step, but insufficient to get the AI safety that we'd want.
Thanks for a really beautiful talk. I'm John Vastala. I'm a postdoc at Harvard. You started with bilaterians, but even single cells are pretty complicated, and there's some evidence that they can do very simple forms of associative learning. So just wondering about your thoughts as to where that fits into the overall story.
Love that question. So I think there's a really beautiful sort of thing in there, which is the same algorithms in nature seem to get recapitulated in completely different means. And that's really beautiful. Taxis navigation, for example, clearly exists in single cells. And so that exact same algorithm pretty much was recapitulated in a web of neurons in a instead of, it's not the effectors of it are not protein machines or cilia on a single cell, but the effectors are a motor system, which are obviously in a large multicellular organism. So the same algorithm can be recapitulated in multiple means. The same is likely true in associative learning. The associative learning machinations within a single cell, maybe some of that stuff is repurposed in the associative learning of neurons, but for the most part, if we think about associative learning as synaptic weight changes, it seems like the same algorithm of learning associations with things is recapitulated at a different level of scale. And so it would be wrong to say that taxis navigation was invented or discovered by the first bilaterians. It was just recapitulated in a different means, different medium.
This is Stefan from Geneva. This may be a silly question, but what do you think is the next milestone? Or in different words, it's like, what are humans not good at? And
oh man,
do you think how
if we persist, people ask me that question. I don't know. I think I think it feels like the most likely thing that's going to happen is we're going to create intelligences that have human-like abilities in silico. And the question of the ages is going to be, how is that going to modify what humans undergo? I mean, humans are already undergoing evolution, clearly. Over the last 100 years, the incidence of wearing glasses is up some crazy amount because the pressure on eyesight has been eliminated through technology. So I don't know what the next breakthrough is going to be, but it feels like the next big intellectual milestone, if you include our AI children as part of our descendants, is going to be done by them, not our biological brains.
Right. We only have time for one more question.
Thanks for a great talk. My name is Better Albena. I'm from University of Pittsburgh in Duolingo. I guess my question is, we're obviously living through a moment of a lot of excitement or hype, depending on your perspective, around statistical models of language being able to simulate intelligent behavior of various kinds, right, in the form of large language models. So I guess my question is, if you wanted to sort of interpret that excitement generously in the context of your framework, right, you could think of it as we sort of cheated by inferring a lot about the world through the statistics of language.
Yep.
I'm curious how you think about this, right? I mean, is the capacity of these things sort of hobbled by the fact that, we haven't gone through this sort of evolutionary process where all these sort of affective systems are also sort of built into how these things work. So I guess I'm just curious for your perspective on some of the recent developments on that front.
Yeah, amazing question. So the breakthrough model sort of the point is to describe our path here. It's by no means to suggest it's the only path to get to the same outcome. So I think LLMs are a great sort of counterexample, because if one were to take this model and say, this is the only path to get to human level intelligence, it would suggest LLMs are doomed to fail from day one. They would never be able to do reasoning and all these things that clearly they show the emergent ability to do. But they do it with astronomically more energy usage. They do it with astronomically more data to learn this world model. So I think I think the question we have to ask is, what do we want these systems to do? And then ask, what's the right way to build them? Tony Zader actually is a great sort of spiel about this, so I'll give him credit on this, which is, if one of our criteria is that we want AI systems to fail in the way we fail, then this evolutionary story all of a sudden becomes more important. So for example, we already have great self-driving cars. The problem is it's unacceptable to us for them to fail in a non-human way. If every 100 billion cars just decides to find the nearest child and drive towards it, even if the safety of being in that car is higher than a human, we still find that ethically unacceptable because it's not failing in a human-like way. So if the sort of objective function of a lot of AI systems is being imbued with these types of human things, then the evolutionary story, I think, might be relevant for AI systems.
Before we thank Max, I want to remind everybody that there is a poster session that goes until 11 o'clock tonight, and after that, we'll see you tomorrow. Thank you, Max.
Thank you.